So yeah, hello everybody. In case we've not had the pleasure of meeting, I am Phil Yeske, the Science Alliance Officer at the United Mitochondrial Disease Foundation. I have to start by thanking Ashley and Desiree for their kind invitation to be a part of this conference. I've been so impressed with the speakers that we've heard today, especially so with the patient families and their involvement in it. It really is a message of hope and inspiration, and I hope that's what comes out of this final session as we start to wrap up the conference. So I've been tasked with moderating this panel discussion on advances in therapeutics, and assuming I can get my slides to advance, we have a really nice set of speakers set up, but we don't want it to just be a series of presentations. We do want it to be interactive. We encourage questions from participants today. We will move through it with brief presentations from each of the panelists, and then we'll try to reserve some time at the end for Q&A. So I get to go first just to kick things off, and I'm going to start probably with what you want to see most. What does the therapeutic pipeline look like for mitochondrial disease? This is an extremely busy slide. No way you can consume it all. I'm just going to try to touch upon some key highlights here. First of all, this is from late 2021. And I can assure you already in early 2022, it's changed. And that's reflective of a rapidly changing clinical trial landscape for mitochondrial disease. And that applies across all types of mitochondrial disease. So within this pipeline, you'll see some indications that are pediatric, some are more adult oriented. You'll see some that are for very specific types of mitochondrial disease. Others are more general across nuclear DNA or mitochondrial DNA. You'll see various stages. Some are very late stage and getting closer to regulatory approval. Others are still in the very early stages. I think the key takeaway here is back in a decade ago, there was one company actively developing a drug for mitochondrial disease. 5 years ago, perhaps 3. Now you see 11 companies on this pipeline, and I've had contact with dozens of other companies in various stages of preclinical planning that are very interested in developing medicines that are targeted to mitochondria and mitochondrial disease. So this is very much a message of hope for the patient community, but it also puts some burden on us to prepare for this as we move into this clinical trial era, as we all want to see that final column you see there, regulatory approval, start to populate. So what are some of the things that UMDF is doing around how do we get drugs to through regulatory approval? We feel strongly that there needs to be a lot of emphasis placed on clinical trial readiness. What can we do? What role can the patient community play within clinical trials? So one of the things we launched in the fall was a renewed emphasis on updating the information available from our website around clinical trials. We've created a way to make it easy for patients to search for clinical trials that may be relevant to them based on the kinds of symptoms they're having, where they're located. Hopefully you can take advantage of this. There's a lot of educational materials there too, because you need to be informed about what it means to participate in clinical research in order to truly appreciate the benefits that will come from having approved therapeutics for mitochondrial disease. That's a very personal decision and one that shouldn't be made by yourself, but clearly in close communication with your healthcare provider. But hopefully the link that I'm showing on the screen provides a nice central resource for you to learn about clinical trials and what's available. What I'm really excited about is what's going to be coming in the next couple of weeks is a web-based therapeutic pipeline. So imagine that slide I shared just before, but in a much more intuitive way that will capture what is the activity that's taking place within mitochondrial disease from a clinical trial perspective. I also want to mention that we'll spend a lot of time talking about clinical trials at our annual meeting. We are, fingers crossed, planning to get back together in Phoenix in June of this year. So I should have put the dates on here, June 8th to 11th of 2022. Again, you see the link to get more information, but all signs are pointing to being able to bring the community back together. And that's researchers, clinicians, patients, and of course, industry as well around therapeutic development. And we'll spend a lot of time talking about clinical trial readiness. Secondly, in order to enroll in clinical trials, typically you have to have a genetically confirmed form of mitochondrial disease. So we felt really important to do more to try and drive the so-called top of the funnel, the number of patients that have this genetically confirmed diagnosis, so that they can participate in clinical trials. So just a few weeks ago, we launched a first pilot genetic testing project that's very much patient-facing. We give the opportunity for those patients who don't yet have a confirmed genetic diagnosis to go to our partner's website, Probably Genetic, enter some symptoms, and using AI machine learning, an algorithm makes a determination, yes, your pattern looks like it could be mitochondrial disease. You're eligible to receive a no-cost genetic test. So this is a very novel approach, and we're really excited to see how this proceeds and how well AI and machine learning can be used to try to identify patients. But most importantly, we feel we're empowering the patient community, share your symptoms, see if you can be eligible for this. So if you don't yet have a diagnosis, I do encourage you to go check out the link that I show here at the bottom of the slide. And of course, we have to thank Zogenix for very kindly sponsoring this program. And we'll look forward to reporting out on that in June at the symposium on the progress we've made with it. How effective is it? That's the question. Lastly, I just want to briefly share with you UMDF's registry plans. We've talked about MitoShare for some time. We've been planning for this for some time, and we're really close to having it ready to be launched. This is very much a home for everyone. And I think you've heard about a lot of different registries, and some of them are very specific to a specific indication. Others may be for a specific purpose. MitoShare is really meant to be the home for all mitochondrial disease patients, regardless of where you are on the diagnostic journey. Here, we'll have many of the same attributes that you've heard from other registry platforms: a place to safely, securely store your data, and appropriately share it through informed consent. So whether it's genetic data, electronic health records, you can have the ability to connect with your clinician and have them be able to see your data there, symptom medication tracking, so more on the support side. We really think it's a nice balance of what a registry can be. So I do encourage you to check it out. And as we prepare to launch, if you visit the link shown at the bottom of this slide, your name will be added to a notification list. And as soon as it's open, you'll get an email to let you know. Clearly, we want to use those data in the most valuable way possible to try and drive the development of treatments and cures for mitochondrial disease. Goal number one. So I'm actually going to stop there in terms of introductory comments and make sure we leave enough time for the other panelists to join, share their comments, and to then move on to the Q&A session at the end. So I'm going to stop sharing and invite Dr. Marni Falk from Children's Hospital of Philadelphia, who's going to talk about precision mitochondrial medicine using preclinical models to identify therapeutic leads to evaluate mitochondrial disease patients in the Mitochondrial Medicine Frontier Program. All right, Marni, I think we used up all your time on just your title.
No problem. Can you hear me?
Yes.
Okay, great. I'm going to share. Okay, terrific. Can you see my slides?
Let me check.
Am I sharing? Maybe not. Hold on.
Not yet. Okay, they're coming up now, Marni.
Yeah?
Yes.
Okay, great. Well, I just want to start by thanking Phil, of course, for including me in this session, and the organizers. It's been such an inspiring day. I think it's really amazing to have the opportunity to work so closely with the families and the advocacy groups, that this is really a team effort, and to bring the community together, the international community. It's such an honor to participate. So Phil already read my title, so I'm going to jump in. I'm just going to share some disclosures. I am on the advisory board for the United Mitochondrial Disease Foundation, Genesis Foundation, as well as several pharmaceutical companies. And I work with biotech and life sciences as a research collaborator, a consultant, and a PI on trials, all focused on developing therapies for mitochondrial disease. We all know that mitochondria are cellular batteries and they make energy. And that's really why we're all here today, to focus on ways that this fails and maybe common ways that we can come up with therapies. What's been remarkable, I'm a geneticist, and the genetic revolution has really elucidated all the different etiologies of mitochondrial disease. And it's been great to split it up, and there's reasons to do so for recurrence risk counseling and precision therapeutics. But I think I've become more of a lumper than a splitter, because at the end of the day, there's certain common biochemical processes and pathways that go awry, and we can start thinking about not the gargantuan task of treating each one individually, but how can we come up with common therapies that will benefit many people regardless of their specific etiology. And so, of course, this is a wonderful paper by Grenier-Gorman et al. From several years back, which shows all the different processes of bringing nucleic acid into the mitochondria. Once you have the mitochondrial DNA, replicating it. What we're talking about here today is how to properly translate it, and so that ultimately you can make the electron transport chain so that your cells can breathe and convert cellular nutrition into chemical energy, or ATP. When we start thinking about this, we realize there's a lot of ways it can go wrong. And again, today we're thinking about ways that this process goes wrong, mainly due to the aaRS mutations. So we are motivated in our research group and our clinical team to understand how to help everybody with a mitochondrial disease, regardless of where the cause is. And so we've worked on, as you heard Dr. Magee from our research team discuss a little bit ago, thinking about ways that we can move a little bit quicker, a little bit more efficiently, and to inform ourselves on some of the key readouts that matter to families. So to do so, we use microscopic worms known as C. elegans that were developed as a model for neurobiology. Vertebrate animals, zebrafish, that are also microscopic in their early stage and grow to be about a couple inches big. We do use mice, and my colleague, Dr. Wallace, is one of the world leaders in creating the mouse models. And we have many human cells. I think we're close to 500 different human cell lines in the group from different mitochondrial disease patients. What do we do with all of this? We start to study, understand how the different diseases work, what's unique about their pathology, and then how can we use either genetic approaches or drug approaches or libraries. So we work with a lot of pharmaceutical partners to help figure out where their drugs belong, whether they're safe in this condition, whereas they might be safe in a normal disease or a normal person who's healthy. They may not be tolerated well in a person with mitochondrial dysfunction. And then we start to prioritize what's the right drug and the right outcome to study it for a given patient. And we've set up a program I'll come back to called the Mitochondrial Medicine Frontier Program to allow us to pretty efficiently track outcomes in our patients and bring therapies to them as efficiently as possible. What we're talking about today, of course, is how can we move a little quicker? So rather than one candidate at a time, how can we do higher throughput screens? So this is a very simple model, just summarizing, what I could talk to you an hour about, which is that we take different genetic stable mutants, we knock down genes, or we use toxins to impair biochemical functions in the mitochondria. And then we study, again, small molecules, different drugs from companies or libraries, or genetic approaches. And we've created we put a lot of effort into creating really a mini pharma to start screening the cells and the models. This shows you the types of outcomes that we can quantify in the different models. So in worms, in fish, in human cells. And human cells are great, as you heard today, when there's a phenotype, but they're fairly boring, right? They live or die, and you can study some physiologic markers. You can convert them to other tissues, which is also terrific, and sometimes even organoids, which is great, but mainly they're useful for survival and growth and fluorescence types markers, as well as some biochemistry that you heard Dr. Van Hove elegantly share this morning. Worms and fish are animals, and so while they're small, they're mighty. C. elegans only lives 2 weeks. Fish live about 2 years. Each worm produces several hundred more worms in about 3 days. In a week, you could have 1 million. So you can really get an enormous number of animals in which to try and make mistakes and fail and find the right answer pretty efficiently. And so you could look at survival, because they only live 2 weeks. If they're short-lived, you could see if a drug rescues survival. You could look at their development, especially important when we're thinking about pediatric diseases, their growth, and many aspects of their activity and physiology. Fish, you can do all of this except lifespan. Some of the animals are short-lived. You heard Dr. Horvath, who actually we are part of a mitochondrial disease zebrafish consortium with 6 other programs where we exchange knowledge and soon fish lines to really understand many aspects of mitochondrial biology and disease. What's really neat about fish is they have organs. And so because some of them do live to adults, like the ears too that she was showing you this morning, we can study the adult phenotypes as well. So there's really value to going across the species, and we call this cross training, kind of like when you're preparing for a marathon, you might also bike or swim or do other sports to get ready. So before we jump into a patient, we really want to understand, can we convince ourselves in different models that there's value to this therapy? As an example, and I'm just going to go pretty high level and big picture, if we have a healthy worm, the wild type here, N2, and we have a mutant here, this is the worm Neil talked to you about, the complex 1 disease, we might know that it's short-lived. It doesn't live as long as it should. And then we can test it with drugs and figure out, we have a robotic system to do this. What makes it live normal again? And then we can dig in and understand why. The transcriptome, the RNA-seq that you've heard about today, the metabolism and flux, the physiology. And so we could really get a little bit smarter about what are the targets and what are the therapeutic leads. We used to do this manually, which is mind-numbingly boring, I guess. You have to hit each worm on the head under the microscope each day. So thankfully, with our colleagues, there are now some automated methods where you put the worms down and you walk away and come back in a month and you have all of your results. Because the longest lived animal would be a month old. Here you can see that you can get a graded survival response with increasing complex 1 toxin, rotenone. This is another very low cost, very quick, 2-scan test where we can see whether animals develop and move or not. And so this is a wonderful screen that Dr. Matthew developed that we use all the time. We did pretty manually over a decade or so, look at every mito disease therapy that somebody suggested should be useful, empiric in the literature. And we wound up out of about 3 dozen that we tested in the complex 1 disease animal that's pretty sick and is very short-lived. We found a good number, I think around 18 or so, that actually rescue the lifespan back towards normal. And when we group these into 3 categories, antioxidants, metabolic modifiers, and signaling modifiers. And this was encouraging to us that there seem to be many ways to treat a mitochondrial disorder. So we took the best in class, glucose, nicotinic acid, and acetylcysteine, and we published last year the results of extensive studies where we showed that if you actually combine the therapies or make a cocktail, this specific combination, here's the wild type worms, here's the mutant worms. The mutants with the combination lived better than normal. So it rescued them, even better than we hoped. And we can see that there was synergistic effect on lifespan, from the 2-way combinations with the triple combination. And we've created flow sorting techniques to look at the mitophysiology really fast, the amount of mitochondria, the membrane potential, which integrates the respiratory chain function, the oxidative stress, and the unfolded protein response. And we can screen through genetic conditions as well as drug treatments to ask what's better. So this is why biologically we think it's actually important to think about more than one therapy. So for example, nutrition is really important, as any mito disease family will tell you. If you can't make energy aerobically through oxphos, your only other choice is glycolysis, and that burns sugar. So it's different types of nutrition are very important to consider. There's a lot of oxidative stress in mitochondrial disease. How do we quench it? And then NAD is the food for the mitochondrial respiratory chain that needs to be converted to NADH. This ratio of metabolites controls hundreds of other reactions in the cell. So it's not trivial if its balance becomes disrupted. So we don't think it's random that this specific combination added a lot of value to our different worm models. We've created now a whole series of zebrafish genetic models using CRISPR-Cas, including with some colleagues in the Mito Zebrafish consortium. And you can see that they're located at different places in the respiratory chain. We even study one Mito translation disorder due to C12 or65, as well as another disorder that affects multiple respiratory chain diseases. Some of these are short-lived, some of these live a long time. What we are able to do, for example, for some of them with stress, their beautiful golden brain, this triangle right here, if you give them a stress, they stroke. Their brain turns gray. And so you can see this pretty clearly. And we can look here how the combination therapy over 4 hours or over 10 hours, either individual components or combined, can actually rescue the problem. And then we can dig in and start to say, okay, the brain is now functional again. What about the lactate? What about the ATP? What about the glutathione levels? So the animals give you an enormously powerful system in which to test the big picture and then the details. And this is really fun. We've been working on animals that live to adulthood. We can swim them. So this is, we can measure just like in people where we do cardiopulmonary exercise testing, we can put the animals in a chamber and measure their VO2 max and their exercise capacity. So we can test whole body function, and we can do a lot of other neat things that I don't have time to show you. We can do MRIs and look at organs. We can look at visual function. We can look at cardiac function. So a lot of neat stuff. We sometimes have fish with cerebellar defects and they spin in a circle. I've only added this because there were some really neat conversations today specifically about this class of disease. When the mitochondrion inside of a cell doesn't work, we've noticed many times for more than a decade that what happens is the cellular stress response is increased. And part of this response in particular has to do with ribosomes. We know very well, this group, that mitochondria translate their own 13 proteins, and that's part of where the problem is in the ARS genes. But we know that the rest of the cell uses cytosolic ribosomes. And a lot of our early work has shown, that there's a yin and yang effect. When the ribosome in the mitochondria is not working, the cytosolic ribosome is working overtime to try and fix the problem. It doesn't necessarily work, and it leads to a lot of stress. And what this has done is it's created a model for us where we've come up with new therapies to quiet this stress. Even if you don't fix this problem, there are drugs that can change signaling pathways or other organelles to quiet the stress in the cell. I just wanted to show you one example that I'm particularly excited about because it's about to go to clinical trial. And here you can see the mutant worms are short-lived. And when we give this drug probucol, which quiets the cellular stress, it makes them long-lived again, to the same degree as rapamycin, which does similar signaling effects. And we can take kidney cells that would otherwise be dead. And when we give them this drug, they have a chance of surviving this terrible insult at lower or severely high doses. And in the fish, their brains, again, I told you, stroke. They turn terribly gray, and then the animals die shortly thereafter. But if this drug is present, when they see the insult, when they're stressed, they can survive it without the injury. And their neurologic function is normal. And then taking the same drug to a mouse, we showed that this mouse model that can't make coenzyme Q, and so it has multiple respiratory chain dysfunction, it gets a terrible kidney disease, and its kidney can't reabsorb things. Its podocytes become flat, and it leaks albumin in the urine. But when we give them this drug, their kidney function can be totally preserved. So we're really hopeful that here I just showed you a drug that quiets stress and worked in different models and different genes. And now we're going to be testing it in people. There's a lot of drugs coming like this, and I think it's an incredibly exciting and hopeful time. Now it's not a matter of will we find a therapy, it's which therapy will we find for which patient and how well tolerated and what combinations will best benefit each disease. And I think the cells will become useful again, as companion biomarkers for these drug effects, but then also to figure out and make sure they're safe before we put them in any patient. So this is what we do. Before we jump to clinical trials, we stop and we go to laboratory animals and cells, and we try to understand where we see the best effect in cells, in the different models, on efficacy and toxicity, and then we can design smarter, smaller trials with people that are likely to benefit and less likely to be hurt by the medicine. And this is my last slide, just to say this is a village effort. Really, I would say a world village effort to provide care. We provide care for patients from most states, about 45 states in about 15 countries. We try to diagnose them, help in their management. continually do clinical research. We've enrolled 1,600 people in our parent study so far. And we have cells, like I said, on 500 different individuals. We do routine data outcome measures on everybody, surveys. We're really honored to partner with foundations to share this data with them and organize national and international efforts. Like there's now a Lee syndrome roadmap project that the international Mito patient community has put together that we're participating in. Again, I think the community is really well positioned now, to harness the information, get the right people into trials, and start to see regulatory approvals, which, as Phil mentioned, is the goal. And I think translational research is an important and critical part of this cogwheel, so that we don't just go with what we think might work, we unbiasedly test it and bring the most potent and safe and effective therapies forward. So to conclude, I hope you'll agree that preclinical animal and cell models have utility for different mito disease subtypes, to decipher their mechanisms and optimize treatment effects on really what matters to the FDA, which is survival, function, and feeling. We can test nutritional components, drug concentrations, different mechanisms, and we prefer a therapeutic cross-training approach to convince ourselves of the best therapies to quickly find, to validate, and to bring forward. And I think that this approach can and should be done for the ARS class of disease, to really see, are there therapies that might benefit everyone, recognizing that, of course, it would be terrific to have individual therapies, but it may very well be that there are some common therapies that would help many of the patients. So thank you very much. This is just a summary of all of the people involved in our laboratory and clinical teams, our funding, including many family and foundation partnerships, and additional colleagues. So I just wanted to say thank you. I have to apologize in advance. I need to leave halfway through this session. So Phil, I don't know if we want to answer questions now.
Yeah, so maybe we have time for one, Marni, since we're a little pushed here. Fantastic presentation. It seems like the key takeaway from your presentation is the importance of translational models and robust translational models. I guess the challenge is, and I'd like to get your thoughts on this, is hundreds of pathogenic genes have been identified, right, that characterize mitochondrial disease, extremely complex. How do we go about prioritizing? Because we have limited resources as well. How do we go about prioritizing which genes to move forward, which interventions to move forward, or groups of genes? Like, what are your thoughts there? Because obviously we just don't have the capacity to be able to move everything through a program like this. Or what are your thoughts?
Yeah, no, I think we're getting to the place where a lot of people are involved, right, due to the awareness and the efforts. I think the foundations, honestly, are the ones that bring light to the priorities, right? I do think, and in my whole career, I've seen it, right? It's a team effort. And so where there's a voice, right, there's typically a response. Where there is knowledge, there's people that rise to sort of address it. So I think it's valuable to go after the genes, for example, where there is resources, because the other problem is that the federal funding, for example, they don't support one rare gene at a time, typically, right? And so it's very hard to do that in an efficient way that will change the lives of the people living today with the disease, not in the grand future. So I think the speed of the resources and the prioritization, and then the communication, right? That it's not just somebody from a biobank, but that you're communicating, you're assessing the families, you're understanding their needs. I really think the way to move this forward is where everybody's going in this group, right? It's really the collaboration, the connectivity, and the continual keeping it in the spotlight and bringing people together. So again, I found today incredibly important and invigorating. I learned a lot. I was really grateful to participate.
Right. Well, thank you for your comments. Thank you for your contributions. We're sorry you can't join us at the end of the session for additional Q&A, but we're going to move on to the next speaker now. All right, well, we're going to move on then to our next panelist. That's Chris Moxham. He is the Chief Scientific Officer at Rarebase. And Chris is going to talk about Function, a scalable platform to enable patient-centric drug discovery for rare diseases. Chris?
Thanks, Phil, and thank you to the organizers for allowing me to join. First, I'll ask if you can see the slides.
Yes, we're good.
Great. So Rarebase is a biotechnology company that is focused on discovering therapeutics for genetically defined rare diseases. And at Rarebase, we have a fundamental belief, which is that every patient deserves a shot at a potential therapy. And that belief system is something that spurred both of our co-founders, Ono Faber, someone you heard about in a previous presentation, who himself is a person living with a rare disease and was a co-founder of Allstripes, and is a co-founder now of Rarebase. And he partnered with Omid Karkudi, who is our chief operations officer, to found Rarebase about 1.5 years ago with this simple mission and belief system. And I had the pleasure of after 25 years in pharmaceutical research and drug discovery, to join both of them in December of this year, but even more so, had the great privilege to join a great team of professionals who all share this common vision, both internal employees and advisors. And the company continues to grow in a great way, and we're all pulling in the same direction to ultimately discover therapies for patients, for people living with rare genetically defined disorders. We are based in Palo Alto. We're in the midst of building out our lab in Palo Alto, and this will be a high throughput, state-of-the-art drug discovery laboratory. We are now conducting our research operations with external partners, and that's moving along in a great fashion. And moreover, we believe that we have the opportunity to innovate in this space in two primary ways to really create value for patients. The first being, and you've heard about this, to repurpose approved drugs. There's a real opportunity here to screen approved drugs that have all of the right drug-like properties in different model systems to find opportunities that can move quite quickly into the clinic. The other, of course, is that we will have the capabilities to conduct novel drug discovery, and we believe we will invent novel drugs, as you just heard about, of the like that you heard about from PTC Therapeutics. The other is that Function is the platform that is our drug discovery platform. We've already started to work with patient foundations like the Cure Mito Foundation to survey approved drugs for their properties, that some of which are shown here, and look at approved drugs and start to prioritize and rank order those approved drugs for their potential use in the context of modulating SIRF1 expression, for example. This approach to repurposing drugs is not a new one, and frankly, it's been validated. And I show you 3 examples here. There are many more. Certainly raloxifene, which is an FDA-approved drug that was approved for the treatment of osteoporosis, a bone disorder, and now has been repurposed for the use in invasive breast cancer in postmenopausal women. Rapamycin, which again was initially approved to prevent organ transplant rejection, has now been repurposed into the disorders shown there. And tamoxifen, which back in 1977 was approved as a hormone therapy to treat metastatic breast cancer, 30 years later found it and repurposed it for treating bipolar disorder. So at a fundamental level, Function, the purpose of Function is really to discover disease-modifying therapies that can act on the root cause of these genetically defined rare disorders by modulating gene expression. And in the upper part of this slide, you can see that we have sort of the wild type scenario where there's normal gene expression. There are healthy levels of the protein made. And in this example, the flower has the right number of petals. And in the disease context, you can appreciate that there's a mutation as shown by this pink bar that leads to a reduction either in gene expression and consequently protein expression leads to that disease phenotype. And what we're doing in the lab is 3 primary things, one of which is we believe that context matters and in the context of the screens that we run. And so we are very focused on implementing cell models of rare diseases that really recapitulate that disease state. And so we are using iPSC technology, differentiating these iPSCs into the appropriate cell types, doing this at scale so that it allows us to screen both a small molecule library that we have amassed, as well as utilize functional genomics approaches using CRISPR technology, for example. And we're capturing features from these cell types in response to these drugs that include both high content analysis of gene expression at scale. So we're able to use high throughput 3' RNA-seq to quantify over 10,000 genes at a time in response to each drug. We're also using high content imaging, where we can capture over 1,000 cell features at a time that allow us to build a very rich data set in a data matrix that includes both cell types, different perturbagens or drugs or CRISPR modifications, in combination with these cellular features. And this data matrix will be amenable to harnessing the power of computational approaches using both machine learning and artificial intelligence to not only predict hits, but also ultimately predict patient response. So to take you through this a little bit in more detail, here's an example perhaps in the wild type setting where you've got in a cell, you have a network of responses that lead to appropriate gene expression and essential proteins being made, and a phenotype of a neuron, for example, that has that quote, healthy or wild type phenotype. And we can capture all of these things using the technologies that I described. In the disease setting, either using patient-derived cells or genetically engineering these models of disease, you can see that the gene regulatory network becomes disrupted. There's a lack of essential protein expression, and that manifests itself in a variety of different ways, both at the gene expression level, but also in the context of that cellular phenotype. And of course, once we have this disease phenotype captured, we're then looking for small molecules, in this case, that revert that phenotype back to the healthy phenotype that we've already characterized. And so again, this has been done before. And so we are implementing a variety of different machine learning approaches that we call Hit Finder, that allows us to rank the best potential therapies to move forward. We're also developing something called Pathfinder that allows us to potentially identify alternative routes to achieve the same end, to revert that disease phenotype back to a healthy phenotype. And lastly, Model Matcher, that allows us to enable mapping from model cells in the lab to easily identify biomarkers that could be used in patients in a translational context. So what are we doing this year? So we've kicked off a screen with about 4,000 drugs that include a number of FDA-approved drugs, virtually all of the FDA-approved drugs. And that's going through a high throughput screen to ultimately identify therapies in that context. We're also using CRISPR technology to create about 1,500 different genetically engineered cell models to characterize those, again, using that high throughput 3' RNA-seq approach, the high content imaging that also essentially does two things, one of which allows us to create a baseline from these disease models. It also allows us to plug them into our drug screening approaches. And using either these genetically engineered cell models or patient-derived cells, we can then conduct what's called a clinical trial in a dish that allows us to look at the response to a particular potential therapy across a variety of different patients, if you will, in this in vitro context. And so this is a significant undertaking that's well underway, and we have great goals in anticipation of achieving this in the upcoming year. So with that, I'll stop. I'll leave you with this quote. It certainly, I think, is a fact that we will all be patients one day. And this comes from our wonderful CEO, Ono Faber. And also invite you that if you have interest in partnering with us, to reach out and contact us at the email address shown here. And with that, I'll turn the time back, Phil, to you, and maybe leave time for Q&A.
Thanks, Chris. Really fascinating talk, and lots of questions kind of come to mind that I would love to get into with you. Let's allow our last panelist to deliver his comments, and then we'll reserve a little bit of time for Q&A at the end. So with that, we'll have our last speaker, which is, Paul Kruszka at GeneDx, and he's going to talk about commercial laboratory outreach programs for rare diseases. Paul?
Hey.
Your audio cut out.
All right, let me see here.
I think you're okay now.
Okay, can you hear me now?
Yep, but we don't have your slides.
Okay, let me go back to my slides. I got to apologize in advance. I lost power at my house, so I'm running off my phone right now. So I'm doing my best I can. Can you see my slides now?
Do not.
Paul, if you want to email those to us, we can always display it for you. That may be taking up some of your bandwidth.
Okay, you know what? I'm just going to try to go for it because my computer is just about dead. So, okay, so
looks like they're coming up. You're all set.
Okay, good. Well, thank you, Ashley. Thank you, Desiree, for inviting me. This is fantastic. You guys have done a great job. Congratulations. I work at GeneDx. We're a commercial for-profit laboratory. I'm chief medical officer. I'm a clinical geneticist. And really what I'm hearing today and what I'm seeing outside is that our world is changing. There's so many new therapies. There's 900 INDs for cell and gene therapy in the FDA right now. And we're seeing it. We have multiple collaborations with many therapeutic companies, and it's really exciting. One thing we're also doing is we are moving into the newborn screening molecular arena. We're collaborating with a number of states on some really large projects that we're going to announce at ASHG coming up this year. And so our purpose at GeneDx is to revolutionize healthcare and change lives by unlocking the answers from within. One cool thing about GeneDx is we've done now over 350,000 exomes. In a lot of cases, we have the parents of the exome. That's your entire protein coding genome. And we've been at it quite a while. We're a spinoff from the NIH in 2000, where I previously had worked for the last decade. And we do a lot of research. We recently published in Cerebral Palsy and JAMA and also in Nature. And this is just an advertisement, not bragging, that we are very interested in the research world. Another thing, which is really cool, we do a lot of reverse genetics. We've got variants, and we have very good phenotyping from the providers out there. And although we do not have any longitudinal data on many of the people that are sending in samples, we can do all kinds of principal component analysis to cluster different phenotypes by age, and this is really cool. And so I went on the website for CureARS. It's a wonderful website, and I noticed that Ashley and Desiree are standing in front of the Children's Inn in one of the pictures, and that's at the NIH. So I used to walk by that every day, so that was cool. I'd love to learn why you were there and who you work with. I'm sure our time intersected at the NIH. So I did a screenshot on their video. I went and watched their video a few weeks ago, and one of the barriers to treatment is lack of patient population. And that's one thing we do at GeneDx. We have lots of population, like I said, over 350,000 exomes at this point. We expect to do about 150,000 exomes in the next year. We do about 70% of all commercial exomes in the United States, and we're have the largest clinical repository. So I looked at Mito-aaRS genes. We've seen about 160 cases in the last 5 years. As you all know, this is a very rare condition. We see with the recessive conditions usually a little more rare, but we have a number of cases, and so this is the type of things we do. In addition to working with biopharma, we actually did work with CureARS, and we do send this letter out when we diagnose a case, variant in one of the 18 genes. We do send out this letter so that the family can contact Ashley and Desiree. So here's my email. That was very short. I was kind of rushing through this. I'm afraid I'm going to lose power at any second. Anyways, thank you again for allowing us to be here. It's a real privilege to be here. And here's my email. Certainly, if anyone has any interest in collaborations, any research projects, that type of thing, please contact me. I made it.
Yeah, good job, Paul. Yeah, no, thanks. Interesting to get the overview of GeneDx. I mean, your company has had a long-term presence in the mitochondrial disease community. We're really thankful for all you've done to help with diagnosis of our patients from a time when it was a lot more challenging than it is now. So we appreciate all the efforts from GeneDx. I am going to go ahead and invite the other panelists to turn on their cameras. And again, Dr. Falk had to leave early, so she won't be able to join us for this. So it looks like we have about 10 minutes, gentlemen. A range of talks on a range of subjects, right, in a very complex area, right? When you start talking about developing therapeutics for something like mitochondrial disease. But, I'm just struck by the importance, right, of patients, patient advocacy groups, obviously drug development companies, diagnostic labs, and increasingly companies like yours, Chris, Rarebase. Maybe we'll start with you, that are sort of facilitating right on the edges the development of treatments, whether it be for repurposed drug or novel composition of matter. as you've mentioned, that this approach is not new. What has changed? What has evolved that now makes this approach, right, more likely to be productive and lead to therapeutics for rare diseases?
I think it's a combination of advances in technologies, either in terms of cell modeling, technologies that allow you to essentially interrogate the transcriptome at scale.
Right.
So with these RNA-seq approaches, now you can measure 10,000 genes at a time in response to
these are the sorts of things that used to only be in the top echelon academic labs, right?
That's right. And of course, there's the computational end on the back end, and certainly we're not taking human creativity out of the process. That's important. But the thing I learned certainly in my past experience using this type of approach is that among the 3,700 known root cause genes that have been associated with these different 7,000 rare diseases, with this type of technology, you can survey a very broad swath of those 3,700 genes in one screen.
And I just wanted to clarify, you're not just focused on neurological disorders.
That's
in
correct.
theory,
Right.
You can differentiate these iPSCs into a number of different types of cells.
Absolutely. So either in very intentional screens with cell types, or certainly from the data that you generate in a neuron, you will come across opportunities to move into a different disease space and sort of this disease hopping fashion.
Yeah, well, the hallmark of mitochondrial disease, of course, is multi-organ, extremely complex. So, the ability to have models that cut horizontally right across all these different types of cells and tissues is really important.
Absolutely.
So Matt, is the most tenured, I won't say the oldest, right? The longest tenured
grandfather,
involved in mitochondrial disease therapeutic development. We've had this conversation so many times, but I really think it's important that the patient community hears it, again and again about how, drug development's hard, right? But there's a very clear role that the patient community needs to play, right, in order for therapeutics to be developed. And so finding the patients, engaging them, just talk briefly about, even how things have changed over that decade plus in terms of how patient advocacy groups are coming to bear, or other mechanisms, or in your opinion, are we heading in the right direction towards having more effective clinical trials?
Yeah, so I think as it was in the beginning, as it is today, as it always should be, patients are the center of everything we do. They're the goal. I always tell my teams that this is why we get up in the morning and do what we do. And it's become incredibly clear that developing drugs is a team sport. It's the literal takes a village. It takes the patients, it takes the families to help us understand the diseases, to know how to design drugs, design clinical trials. It's close work with your group, UMDF, the patient foundations, advocacy groups, genetic diagnosis. I mean, all stakeholders, all hands on deck. Right. We're all in this together. We all have the same goal, which is to deliver effective therapies. It's true. It is so different today than it was, 2009 when we treated our first patient. I'm so proud of all of the activity, all the different advocacy groups, the engagements of patients, the engagements and partnerships across industry, academia, and foundations. And I think it's incredibly important if we're going to be successful in discovering and developing and delivering therapies that we really evolve our culture of trial participation and understanding what it's like to be in a clinical trial, what that actually means. I use words like randomized placebo control.
Right.
That's kind of a complicated word, but when it comes down to it, it means, hey, you're going to go in a trial, you may get the drug for a period of time, you may get placebo for a period of time. And being in a trial is different than just going to your doctor and getting something that might help. And it's sometimes, and we know it's an enormous sacrifice for patients and families to participate, but it's so incredibly important. And I think evolving the culture of clinical trials means a lot for us, for a company too. We've got to learn a lot. Right. The doctors who are clinical trial investigators, they need to learn a lot. And it's this idea of community that we all are working together and growing. And I just think we're in such a, as you said, Phil, such a great position and has so many trials and so many opportunities to help patients. And because we know it's not going to be one therapy, the more, the better for all patients.
Well, you know, I'm really happy to be able to show a slide that, you know, with 11 companies on it and, you know, many of them actively recruiting for clinical trials. We also have to acknowledge we've gone through a pandemic for 2 years now, right? That it really has put a lot of pressure on clinics, on clinical trial recruitment. We're seeing the light at the end of the tunnel. I think everybody is ready to kind of get back out and participate, but you know, that's just another place where we're really going to need the patient community to kind of rally around the opportunity that is there, because sometimes these windows are only open for you know, a relatively brief period of time, and we'd hate to let that go by and set back the entire field for years on top of that. So thanks, Matt, for those comments. I do want to encourage the meeting participants. If you have questions, we don't have a lot of time, but let me try to maybe grab one from the Q&A. And Paul, this is actually for you, if you're still connected, right? How many of these 160 cases have both allele pathogenic variants versus how many have at least one that is listed as a variant of unknown significance? And maybe that's an unfair question now that I've read it.
I haven't looked at all of them. I had Renqui, who I'm sure you know, Phil, give me the data. But I looked at a few. I did look at a few of the case reports, and they were both pathogenic or LPath, the ones I looked at. It also, what's interesting is the different ages they presented. I saw rs2, that was a 38-year-old man that presented. So it was interesting. I do enjoy the case reports, but I couldn't read all 160. The ones that I looked at were LPath and PATH. But I can find out for you, Dr. Van Hove. Certainly my email's there, and we can connect, and I can give you a more accurate list.
We can definitely make that connection after the meeting. You know, I think, Paul, while we've got you kind of on here, obviously GeneDx is focused on genomic data, right? Testing these patients, generating data from it. I think we've heard this, though, in a number of the talks. It takes phenotypic data, genomic data, electronic health record data, clinical trial data, you know, this sort of a lot of different data types to be successful ultimately in developing therapeutics. I noticed that, you know, GeneDx is moving in this direction too, becoming more of a broad, you know, data company, right? You know, going forward. Where do you see genomic data fitting with other data types in terms of facilitating drug development? Especially that you might have access to, that we can really help drive research.
I'm sorry, I might be breaking in and out. We require a lot of phenotypic data because our algorithms require the phenotypic data really to make our call. So we abstract all the HPO terms, the average of 11 to 12 HPO terms for patients. So we get 30 to 40 pages of PDFs from most people. So we abstract all that. The problem is we don't have longitudinal data.
Right.
And most of our patients are very young, or these are infants and children. They really don't have much of a history. We're doing a lot of NICU sequencing nowadays. So, we've got 30 days of life of history. So we unlike adults, we don't have that long history. Usually we don't have longitudinal data, but we take it very seriously. We do abstract it. We demand it. We want it because it makes our algorithms better.
Yeah, and so for our patient families, again, longitudinal data, particularly important for developing the natural history of the disease, right? How does the disease progress over time? And that's incredibly important to drug developers and how they're designing their trials, researchers and understanding or making short-term gains or not. So a really important part of it. Paul, I think we're going to let you put your arm down.
Okay.
I think you're running out of bandwidth there. And I see we're at the top of the hour, but Chris, I'm going to give you the final word. Patient centricity is sort of this buzzword, if you will, right now in therapeutic development, particularly for rare disease. Talk about it like from your perspective. How does Rarebase fit into that? How is it different? How important is that, right, to companies like yours in being successful at bringing therapeutics to rare disease?
Yeah, well, first of all, it's absolutely critical. And what really is, I think, differentiating at Rarebase, and having spent time in pharma and biotech, at Rarebase, the patients are engaged with us from the very beginning.
Right.
not just in terms of understanding the natural history of their disorder or collecting biosamples. They're actively seeking to fund the screens that we're running. And so they are very much, in a way, steering the drug discovery that's occurring at Rarebase. And I think it's a great partnership. And I think it is differentiating from other, simply venture-backed biotechs that would not spend their time repurposing FDA drugs, approved drugs. And that really, I think, is differentiating at Rarebase and an important component to really bring therapies to all patients.
Well, as a Mandalorian would say, this is the way. There's no way we will get to it unless we all remain focused on the patients and put them first, put other agendas aside and what's best for the patient community. So I thank all of you for your contributions, your comments, and I think with that, I'm going to turn it back over to Desiree and Ashley to wrap up the conference.
Thank you, Phil.
You're welcome.
I would like to actually introduce Brent, who is the Executive Director of the CHOC Children's Research Institute. He is going to provide our closing remarks, and then Ashley and I will have a short message to say after him. Welcome, Brent.
Oh, thank you very much. Wow, what a day. It's really been a pleasure. I'm Brent Dethlefs, Executive Director of the Research Institute at CHOC Children's. I've been at CHOC for almost 26 years now. It's pretty hard to believe. I like to tell the story that when I came on board that we were and hired our first research administrator, that we were given a shoebox and told that everything research was in that shoebox. So from there, to over 530 active clinical studies, human subject studies. It's been quite a journey. And I like to think of myself as a recovering molecular biologist. So having the opportunity today to put my administrator hat aside and listen to all this has really been terrific. I really do want to thank the founders of CureARS, Desiree and Ashley. Without you, we would not be here, and certainly your children, Daphne and Aubrey. We developed with under leadership Dr. Terry Sanger, who's our first ever VP of Research and Chief Science Officer, has been here just a little over 2 years. And one of the first things that we embarked upon was a strategic plan. And in that plan, one of the tenets of the plan is begin with one child. And certainly in the area of rare diseases and ultra rare diseases like ARS, seeing one child, learning what the challenges are from that child and moving forward is absolutely important. Coming from an academic background for many years, we used to think of translational science as from bench to bedside. And for those of you who work in academic environments, you know how long that takes and how seldom it happens. So here really, we believe that it's really bedside to bedside, that we have the extraordinary opportunity at CHOC to learn from our patients, that the challenges we see at the bedside are really the ones that we should be pursuing in research, and we are certainly prioritizing that. In no small measure, because of the strength of our metabolic team, Dr. Abdiner, I want to thank him very much for his leadership in the conference today, as well as our organizers, Nadine and Kevin and all the rest of the team. It's really been wonderful today. But as part of that strategic plan, rare diseases is a focus of research at CHOC. And at the heart of that is really community patient-family partnerships. So it really warms my heart to see all of you here today. We envision one day having a campus with a big family conference center where we can all come and, Lord willing, and the creek don't rise, that the pandemic eases and we can all be together, not on Zoom, but to see each other face to face, that we will have that one day. But in the meantime, I just really want to commend the efforts and thank you all for allowing CHOC to host the group today. We may be here today talking about science. It's really, again, for those of us who are into this, just wonderful. And I'm sure for the patients and family, a wealth of resource, but it's really love that brought us here today. The love of two mothers for their kids and the commitment of those mothers to make a difference. If you see something, you see a problem, do something. The truest form of belief is certainly in action. And I believe if we stay disciplined and focused and our eyes on the prize and we work together, that in the long journey to make a difference in the lives of kids with ARS that love will find a way. Thank you so much for today and for allowing all of us at CHOC to be a part of what has been just a terrific conversation.
Thank you, Brent. So as we've heard today, there's a ton of exciting research being conducted on the ARS family of mitochondrial diseases, from the fundamentals of molecular pathways to the study of yeast and animal models to clinical presentation and treatment. Our presenters today covered the gamut.
Our hope in putting together the symposium was to create a dialogue between the various ARS pathologies and provide opportunities to discuss future directions of research. Based on the enthusiasm we saw during the panel discussions, I'd say we made a big step in that direction today.
We are representing the 19 ARS2 genes, and we tried to provide representation from all the communities being studied. We plan to do the symposium annually to provide updates on the current presentations as we expand. And we will post this video on our YouTube for sharing. Do not hesitate to reach out to us for inclusion in next year's symposium.
Thank you to all the speakers for taking the time today and the audience for the insightful questions. We look forward to continuing the dialogue at future remote and hopefully in-person meetings.
And just lastly, if you're interested in submitting a proposal for funding, please email us at cure4ars@gmail.com. And everyone stay safe and have a good evening. Thank you for joining.